All right, everybody, thank you for coming along today. Today we have Andy, who's been keeping us in suspense about the topic and name of his presentation up until right now. So I'm just going to read it off. He's talking about understanding athletes at a molecular level. About chance.
Thanks for that.
Over to you, Andy.
The reason I kept you in suspense is because I've sort of written this morning. Not too much time to do that. So obviously it was brought forward, so I figured it'd go. I've also not presented sort of proper science for a while, so I'm no doubt a bit rusty. So I figured, just in case, this is essentially what the people want, isn't it? So living in Australia, that's what I've learned. Doesn't matter how this goes, as long as you've got a nice picture of a dog, you should be okay. The other thing I'd like to let you know is we are not the same people. We're very clearly different. So Stephen Merchant, me, and Edward Snowden, whilst there is passing similarity, I do acknowledge we are not the same. So I'm not a well, maybe I am a fun-loving criminal, we don't know. But all right, so a little bit about my background, which you all know anyway, but we'll go through it. So freezer to furnace. So essentially, obviously, I'm from the UK originally, from London, grew up in Wimbledon. And then I bounced around sort of England and Australia from then on. So started out, obviously, grew up in London, went to Adelaide twice, that didn't quite work out twice. Then eventually we tried Perth. So I went to Guildford Grammar over in Perth, which was the same school that Keith Ledger went to. He wasn't there when I was there because it can't exist at the same time. Thereafter, I moved on, obviously, through back to Perth. So I did my undergrad, my schooling in Western Australia. I did my undergraduate at the University of Western Australia. Thereafter, I did a PhD at, where did I do that? That's a good question. At ECU and in the AIS. But in the interim, I also did a graduate diploma of education at UWA. And I suppose a little bit of background as to why I did that. When I did my honours, I didn't have a good experience in honours, and I figured research is the devil, and I didn't want to ever do research again. So I took a slightly different route. I figured I like coaching, so I'll go and do teaching. Turns out I didn't really enjoy teaching screaming children from year 9 onwards. But university seems to be okay, so we've moved on to that. I then did an internship at the AIS, which I don't shut up about, so you all know that anyway. Then I did, obviously, the PhD, as I mentioned. So that one looked at iron supplementation in athletes when they train at altitude, predominantly distance runners. So we had a great time doing that. I then moved, as you can see, I bounced around between Canberra and Perth, then moved to Bedford. So Bedford is in the middle of England, all right? So it's 45 minutes on the fast train outside of London, or about 3 hours on the slow train, as I found out the wrong way once. Never did that again. And that used to cost £45 per day to get there, and then another £45 to get back. Did that for a couple of months at the beginning of my job when I was working at the University of Bedfordshire. And that wasn't too fun. Thereafter, I moved to Östersund in Sweden, where I was working at the Swedish Winter Sports Research Centre. And eventually I got fed up freezing my ass off and moved to Melbourne, which I'm not sure if that really has fixed that. It's still cold and ridiculous. So now, speaking to Anthea, predominantly, a lot of the time, she was like, you seem to have the same profile picture forever. You always seem exactly the same. So here's proof that I don't look the same, although I'm not sure I'm super convinced there. But you can see in school, I was kind of, fat and could run a little bit. In undergrad, that's a little bit of a rebel, a bit of sausage in the hand. Through my PhD, I focused more so on running. All right, so happy with that. Used to do well. Then my postdoc, there's me literally not able to feel my hands anymore, trying to take blood lactate from cross-country skiers. And I basically had to give up that day because my hands actually froze. All right, well, that's kind of my background. So we'll go on to the talk proper now. Now, you're aware that I do some kind of research around metabolic phenotyping and metabolomics. That's what we're going to talk about today. But I thought I'd give you a bit of a background as to how I actually got into doing that. So I've named this sort of the 2 Mile High Club. So it really came about during my PhD. So I did my PhD, as I mentioned, obviously at the Australian Institute of Sport. While I was out there, we had another researcher there, Nathan Lawler, from Murdoch University at the time. Ironically, is now at Murdoch again. But he had an idea of doing basically some metabolomic research looking at blood doping. I needed some extra cash to get my third study done. because we didn't have that fully confirmed. And he figured, well, if they kick in a little bit of cash, then can we get this 2-week altitude training study done at the AIS? I was like, sure, why not? And we actually have taken what is still actually the only research paper out there of live high, train low from metabolomics perspective. And I'll explain what metabolomics is and why that's important later on. And this actually quite fascinated me. So I was discussing this with my supervisor at the time, which was Chris Gore at the AIS. He was head of the AIS at that time. And, you know, he really encouraged me there that this is probably the way of the future. And it's something that maybe we could pursue long term. And so, you know, I put that down to my little black book of ideas, which was on ice for many years until I actually got a research position, enough cash to actually do any research in this area. But here we are. So today we can talk about athlete profiling and athlete monitoring. So with athlete profiling and monitoring, but predominantly profiling, it's almost like an incomplete puzzle. So you can see there, you know, we're always trying to look through the window as to what's actually happening in an athlete. And so here, as I've got, it doesn't always look how we think it might look. So you might have an athlete who's competing really well in kind of one area, and you've got an idea of what they should look like, maybe metabolically, physiologically, biomechanically. But as we know, it looks kind of deceived here. For the psychologists in the room. So being a distance runner, I was always fascinated with the 800 meters, mainly because I absolutely hated the 800 meters because it's a breakneck fast kind of distance run. It's really a joke on endurance runners because it's almost like a sprint, but you got to carry on. So looking at this here, we've got two different, extremely different athletes here, both incredibly successful in the 800 meters, but for different reasons. So on this side, we've got Alberto Juantorina from Cuba. So he was a 400 meter, 800 meter runner. So as you can see there, he's got an amazing 400 meter. So he actually won a gold medal in the 400 in 76 at the Olympics. And he also has won a gold medal in the 800 as well. And then you've got Seb Coe. So Lord Sebastian, he's got every honor under the sun, it seems. Seb Coe. So he had a pretty good 400 meters. I mean, 46, I'd take that in a heartbeat. But then also he was the world record holder for 800. Ironically, he never won a gold medal at the Olympics in the 800 meters. So he medaled in the 1500 meters, which wasn't his event. And the story goes that the other person that he was main rival, so that was Steve Over, another Brit, he actually won the 800 meters. Seb Coe won the 15 when it was meant to be the other way around. So why is it then that we've got two of these athletes here? They're built different, but they can both do fantastic 800 meters. So you've got one person who's stepping up a distance, but is actually pretty quick. Then you've got another person, another athlete here, Seb Coe, stepping down a distance from 1500, all right, then able to do sort of a good 800 as well. Now, common sense would tell you they're built different, right? They shouldn't train the same way. So why is it under the hood they are built that different? And this is the kind of thing that always fascinated me to try and disentangle that information. So any of you who are into swimming, which isn't me, but we're in Australia, so we can't avoid it. Basically here, sprint swimming training, are we doing it wrong? So I've had a good long chat with some of the people over at the AIS, the VIS, and various other things. They keep asking me about this fellow over here, which is Cameron McAvoy. So Cameron McAvoy is a very smart swimmer, right? He's got, you know, sort of advanced schooling in physics. So he's a bit of a thinker. And this actually isn't rocket science in other sports, but seems to be in swimming. But basically here, he's kind of put in, now the dogma over, or the historical kind of way that swimmers tended to train was they would do a lot of volume. So that was true if they were a sprint swimmer or whether they were a distance swimmer. For some reason, swimmers seem to do a lot of miles or kilometers in the pool. Now with Cam McAvoy, he said, well, why are we doing this? If the purpose is short and sharp, what's the point of putting together a lot of volume in the pool if I'm a sprint swimmer? My event only lasts for a fraction of seconds. Should we just cut out the fat and do it short and sharp? Seems like common sense to me, but that's a question that's worth asking. So this is kind of the fundamental underpinning as to what we're doing. It's sort of my work and Lane's work as to what we're looking at from a metabolic perspective. So we're trying to disentangle really, not so much just sprint swimming, but what's actually happening under the hood physiologically for an athlete when they're training. So we go through a little bit of physiology here. This might look a little bit daunting, but I've just put it there to frighten you all. Basically, right, so athletic training, generally what we're trying to do, whenever we train, we have some form of stimulus. So that stimulus is going to activate a whole signaling pathway cascade. All right, and that signaling pathway cascade is going to try and change some form of gene expression. All right, so we've got transcription, so that's pretty much coding the gene, and then we've got translation. So eventually, that gene is going to have to be translated into a protein. Subsequently, that protein is going to go through and it's going to act upon the target cell or the receptor site, and it's going to enact a change in the cell. All right, and that's essentially the signaling cascade that we would have. Eventually, we do this long term, we should see changes in the athlete performance. All right, so you might see little signaling gene pathway cascades looking like that. All right, looks pretty daunting, where we've got over here what's happening with endurance. And for endurance, it's generally all about AMPK. And then for resistance training, it's usually things like mTOR, although they interact. Now, I recall a bit of a conversation I had with Chris Gore when I was at the AIS doing my internship out there. And we were speaking about metabolomics, we were speaking about gene expression. We always just get really frustrated that we would see a lot of these physiology research projects. There they go and take muscle biopsies, or they take blood, they go and, sort of plot little dynamite plots with the Western blots that they've done. And they go, oh, we've trained, gene expression's increased, happy days. That means we're going to get a better athlete and they're going to perform better. They're going to go and win the Olympics. And in a nutshell, that's pretty much nonsense, because that's not really what happens. So more recently, we've seen David Bishop and friends here basically come out and say, this is from skeletal muscle biopsy perspective. So here's our typical traditional dogma that we've got. We might have an acute training stimulus. We see an acute upregulation in gene expression, but the gene expression itself doesn't mean to say that we're actually going to see a change in the athlete performance. It just means the gene expression is increased. Furthermore, in this paper, they argue that not only is it that gene expression might increase or decrease, that may or may not translate into a change in protein expression or protein changes, and it might not actually then change performance. So I'm going to go through a bit of a concrete example of what I mean here. So my background being altitude, all right, we've got in normoxia, generally we see, so that's this one here, we've got the body's master regulator of hypoxia, which is known as the hypoxic inducible factor. So HIF, as it's known. There are 3 isoforms of that, HIF1, HIF2, and HIF3. They do different things. Now, under normal conditions, generally we don't see the stabilization of HIF for long enough, all right, for that to actually then go and act upon the body in any sort of meaningful way. However, when we're in hypoxia, we get the stabilization of the HIF gene. The HIF gene, so that's HIF1 alpha and HIF2 alpha, that will then go and stabilize. It will then combine with HIF1 or 2 beta. Eventually, that means that HIF is going to be expressed more, so that's the gene. Later on, this will then go and enact a protein change. All right, now the protein change that we have depends on which isoform that we've activated. Is it HIF1 or is it HIF2? Cut a long story short here, they're both activated in hypoxia, both HIF1, HIF2. HIF3, which is the 3rd isoform, helps to shut off HIF1. But what does it do? All right, so if we have a look at what's on the screen here. So EPO, who's heard of EPO here? I'll put your hand up, because that's what all the cyclists, endurance athletes are cheating on, etc. All right, so endurance athletes love EPO. The reason for that is EPO is produced from the kidneys or an extreme hypoxia of the liver as well. But basically, we get EPO that is released from the kidneys. Once we've got the stabilization in hypoxia of HIF2, EPO will then go on and act on the bone marrow, and that will increase the rate of erythropoiesis. So erythropoiesis is the production of more red blood cells. This is from an acute study where they mapped how responsive EPO is. How long does it take to actually stimulate the production of EPO? So here, I've done a maths view, it's 4.5 hours, all right? So we see 4.5 hours of continuous hypoxic stimulation before we see a meaningful increase in EPO concentration within the blood. EPO will then peak about 2 to 3 days after continuous hypoxia for about 14 hours a day or even 24 hours a day. So it takes a couple of days for EPO plasma concentration to peak. So EPO is a protein, right? That's going to do stuff. Whereas HIF, coming back here, that's a gene. That will enable the cascade to happen, but it's not actually going to increase your red blood cell count. Here's another graph. So this one here is actually hemoglobin mass. So this is the end product. So once EPO has acted on the bone marrow, we've got an increase in red blood cell concentration, or hemoglobin mass as it's typically known. Assuming we've got enough iron in the body, we should see this increase in hemoglobin mass that's going to occur somewhere in about 12 days after we've got that continuous hypoxic stimulation. So my point here, even though I've taken a while to make it, is look, we have a think about the time course here. We've seen an upregulation of HIF. So HIF has increased. So that might happen rapidly, but it's going to take probably around 4.5 hours for EPO to increase. Thereafter, it's going to take probably about 2 or 3 days for EPO to max out in terms of its concentration. Thereafter, it has to then go and act on the bone marrow. Then and only then, sort of about 12 days later, we're actually going to see an increase in hemoglobin mass. Hemoglobin mass, if we see that increase, will increase the oxygen transport component in the blood. It means that you should be able to perform better in endurance events. So the point I'm making here is just because we see a change in genes or an increase in, say, HIF1 expression, doesn't really mean anything. All right, so it has to have context. So this brings me then, whilst I got annoyed with that, this brings me to trying to understand more so what's actually happening. So there are many different layers of really the systems biological view. So if we look at the central dogma of systems biology, then we've got, at this level here, we've got genomics, which we all know genome sequencing for COVID. I didn't want to bring COVID up, I'm gonna, because you know what that is. All right, so we've got transcriptomics, so that's what's happening at the transcript level. We've then got proteomics, what's happening at the protein level. Later on, as we carry on down, we've got metabolomics, which is happening at the metabolite level. Now, the reason why metabolomics is of more interest to me is because metabolomics is the closest thing we've got to the phenotype. So that means what is actually happening in the blood, right? So if you go and do exercise, for example, you might have that gene change, you might have a protein change, but actually right at the very end point of that, you're going to get a metabolite that's produced at the end process of metabolism. And that's what we can measure either in the blood, the urine, saliva, sweat, tears, whatever, you name it, we can do it. All right, so this is what I'm interested in, the end point. All right, we're going to come back to swimming for a bit. Figured I had to throw in Paul Gaston's little energy process diagram, although I've changed his aerobic to oxidative because generally we've got oxidative metabolism now. So you're all aware that we've got 3 different main energy pathways. I mean, this is simplification, but we can do it. So any high intensity kind of event we've got lasting a very short duration there, we've got the phosphogenic system. Thereafter, we've got something like a 400 meters for about 30 seconds-ish. Then we've got the glycolysis pathway that's active. And then the main predominant energy system that's active when we go longer than about 400 meters, that's the oxidative system. All of these interplay together. So none of them actually works in isolation. They are always all active, but it's generally we can dial up one or the other one depending on the intensity and the duration of the exercise that we're doing. So when it comes to a profiling perspective from swimming, what they want to do is actually understand each one of those energy systems in a particular way. So ATP, glycolysis, and the oxidative phosphorylation. So we need different tools in our toolbox to try and evaluate that. So this is straight out of the Swimming Australia playbook from the VIS. So here at point 1, they're pretty good at evaluating things like the maximal steady state. All right, maximal metabolic steady state. So things that are analogous to that, something like lactate threshold 2 or critical velocity in swimming. Economy, so that's the amount of oxygen that is required to do the movement. Buffering capacity, that's quite easily measured in the blood. VLA max is something that they're putting in there. I'll discuss that in a moment, explain what that is. Then they've got the anaerobic components here, which is more the VLA max. And then this D prime component. What D prime is generally the amount of work that you can do above the metabolic steady state. And finally, they've got this concept which I don't like, which is anaerobic speed reserve. Now, if you're not familiar with the critical power, critical velocity, critical speed model, it's pretty simple. Basically here, you can do a certain amount of work underneath your metabolic steady state. All right, so you've got this finite kind of thing. If you're exercising at a low exercise intensity, so we call that in the moderate or the heavy domain, all right, then it's generally able to be sustained for a long time. If you're exercising in the severe exercise intensity domain, then you can't sustain that exercise intensity for long. We have this curvilinear decline that we'll see in the amount of work that we can do or the distance that we can actually achieve in that time. So this is almost like a battery. We've got a finite amount, but the rate that you deplete your battery really depends on how hard you go, if you like, or in our case, if you've left the iron on whilst you're watching TV, and you frantically sprint home and hope that your house is not on fire. The other thing then is to note that each one of these are the same size. So depending on how we choose to expend the amount of work that we've got above the maximal metabolic steady state will dictate really the speed that we can move at. Now in sprint swimming, there's probably around sort of a 10 seconds of 60-second effort. So you end up with diagrams that look like this, and they look a bit confusing, but when you actually sit down and think about them, they're pretty cool. All right, so if you look at this, obviously the phosphate creatine system in a 10-second all-out sprint, that's our main guy that's active over here. Eventually, we're going to see some increase in lactate, which is this red curve over here. When we change over to a 60-second all-out sprint, we still see that depletion of phosphocreatine. Yes, we've got lactate that's involved here. All right, but it's a little bit less so, and we've got more of a contribution now for VO2. So that's going to be more oxidative in terms of that energetic pathway. Now, I thought I'd just describe the VLA max part. So with the VIS, they're obviously trying to work out whether these athletes can go and train. So sprint swimmers, are they ready to train on a particular day? All right, so do they have the ability to go and do high intensity work on a particular day? Are they recovered? Are they adapting well? How can we evaluate that in a way that's really to achieve at the pool deck? So generally, in this paper, which was in cyclists, not swimmers, they did 3 all-out sprints. So they had a look at 0, 15 seconds, and 30 second sprints. Did this 3 times over, which would have been brutal if you were in that study. But basically, they then measured the blood lactate concentration. So you can see the curves here. This is every minute, all right, until the curve is really kind of stabilized. And what you'll see here basically is that that peak is achieved in about 3 minutes. So what they're thinking is to evaluate whether somebody's ready to do high intensity interval training or like the zone 6+ kind of, zone 5+ kind of work, so high energetic work. Then you can go and give them something like a 3 by, in their case here, what was the highest one? Should have labeled the axis tonight. I think it was from memory, it was the 10-second one. Basically, they can use that, measure about 3 minutes after exercise to work out the lactate concentration. That gives some indication as to whether the athlete is ready to train high intensity that day. All right, I'm going to criticize swimming a little bit here. Having worked with swimming a little bit, it's a little bit, what would I say, archaic in the way it does things. Stick it in nice and clearly there. So basically, they've got some pretty difficult tools, or I should say like old school tools, to evaluate how an athlete's performing. So historically, they've used things like the Lactate Pro, stopwatch heart rate monitors. Now they've put in heart rate variability. They've got athlete monitoring systems, etc. Problem is they've had this for the last, I mean, I've been involved in swimming since about 2011, and they're still almost doing the same thing. There doesn't seem to be too much innovation going on. So I question why that's the case. Now, the thing is, obviously they came second in the last Olympics, and they had one of the best Olympics ever. But in order to either beat the Americans or stay where they are, you've generally got to try and innovate. The arms race is on really on getting better approaches. All right. So just to prove that I have actually done some swimming testing, even though I can hardly swim myself, there's me as a young, fresh-faced physiologist there trying to frantically write down all of the stroke rates, times, counts, all of that. And then there's me and Lachlan Mitchell, who's now at the VIS, sampling some athletes. And right in the background there with the bald head is David Pine actually doing some work for once. So. Now, if we want to understand what's actually happening in an athlete, we need to go and sample either their blood or another kind of tissue. So one way we can do this is using a muscle biopsy, and that's probably the best way of doing it. Now, the problem there is that you've got this gigantic needle using the Bergstrom technique, and Matt Barley will tell you it's not the most favorable one. I'm sure your Alana's pretty too many of those. Now, you're not getting that muscle back once they've taken it out. It doesn't tend to grow back, all right?
They did ask.
Sorry?
They did ask. Doctor.
Whether it comes back. Doesn't come back. Once they've jagged it out your leg, it's not like it grows back the next day. You just have a hole in your leg. How many athletes can get that done regularly, routinely? Probably not. I wouldn't be too happy with that if I ended up with about a third of the gastrocnemius left by the end of the week. So we need better ways of working out what's going on under the hood in an athlete. So there are two main methods that we can use. One of them is metabolic footprinting, where we collect urine, sweat, saliva. So these are more the end products of metabolism, but more excretion mechanisms. Or we've got the general metabolic components here, which are fingerprinting. So here we can use blood, that's whole blood, or we can use serum, or we can use plasma, or we can use all of them. So the way we do this, we generally collect the tissue sample, extract it, break it up, put it into that kind of biofluid situation, chemically analyze it, and then reduce it down from a data analytics perspective. Spare you the minutiae on how that's done. Now, conversation I've had. So we see a lot of good research done in metabolomics space in skeletal muscle. The problem I have with skeletal muscle is that we can't take that routinely. So I think if this is going to be a useful tool for an athlete long term, then we generally need to have something that's easy to access. Problem with venous blood, in my view, is a lot of athletes don't like having a venipuncture done. To do that on a daily basis, you generally take quite a lot of blood. Typical venipuncture is between 4 to 8 milliliters. We've got all these newfangled techniques that we can use now. So we could take a capillary blood sample. That might only be 1 milliliter or even half a milliliter. And now they've developed other microsampling techniques like touch-activated phlebotomy. So the way this works, you put a little device on your deltoid, press the button, a little microblade will go and sort of puncture your deltoid muscle. There's no nerve endings really in your deltoid. That's why you can punch somebody in the deltoid, it doesn't hurt that much, don't do it, but it can then suck up the blood, all right? And that means you can go and analyze the blood. Now, WADA have just implemented this. So this is in use at the moment for anti-doping purposes. USADA and WADA, they're using that, certainly in the UFC and also in Major League Baseball. Another one that's been implemented already by WADA for anti-doping purposes is the dried blood spot. So this is a micro sample. It blots on a piece of paper. You can then send that in the post with a special secure device. It can then be chemically decomposed, and we can then flag somebody for an anti-doping purpose if we want. And then the final one is volumetric absorptive microsampling, or VAMS for short. So all of these are volumetrically accurate, and it means that we've got a well-known quantity of blood that we can decompose and then analyze the metabolites that are in that to work out what's happening from a metabolic standpoint. Now, while these methods are available, if we have a look at what's going on from a molecular monitoring perspective in swimming already, I've used the first two of these, and I'm sure, Matt, you're aware of these as well. You've used them as well. So Lactate Pro 2, that's bread and butter on the pool deck. We've then also got an iStat, which is used less regularly. In fact, we've got 2 here, and I think we've used it about 3 times in the last 6 years or something. Don't tell Paul, but yeah. But basically, the way that works is that can do quite a lot of metabolites. It's like a mobile metabolite monitor. You can buy different cartridges depending on the panel of metabolites you're interested in. You only need 95 microliters. Put it through, and it's pretty rapid in terms of its turnaround time, so 120 seconds. Here you can get 10 to 20 metabolites. Lactate Pro, it's only lactate, so that's only 1 metabolite. Whole blood for both of them, finger prick. Both of these are already implementable on the pool deck. Now, contrast this with metabolic phenotyping. So obviously this is brand new. We're going to take a small volume of blood, about a milliliter or 500 microliters. We get away with 500. It gets spun down, right? And we will have basically separated out into the watery part of the blood and the red cell part of the blood. We can discard the red cell part of the blood. We only need about 290 microliters to about 200 microliters. And we'll get 1,000 plus metabolites. Now, The reason why that's important is that means we can map full energetic pathways. So all of the intermediates in the glycolysis pathway, oxidative pathways, and also any kind of those phosphogenic pathways or amino acid metabolism as well. So this gives us far more information, but the limitation at the moment is this takes months and months, sometimes years, depending on whether they choose to analyze it or not, and the method that they choose to actually get back actionable results. So it's still a work in progress, but it basically has, I think it'll be the future, let's put it that way. All right, so back to our sprint swimmer. So how can we use metabolomics or these sampling methods, microsampling, and also, you know, sort of those combined energetic pathway understandings to try and get a better view of what's going on in the sprint swimming environment to try and answer Cam McAvoy's question. Now, I had a good chat with the swimming physiology group at the AIS, as well as Tim Kelly, from the R&D officer at the AIS as well. And I coined this term, which I'm claiming, so don't steal it, but basically it's probably been done before, but basically swimming is a training suit. And what I mean by this, if we think of the typical sprint swimming kind of environment, we've got high volume, usually in the warm-up, about zone 2 work, so that means it's under the first metabolic threshold. They then do a main set, which might be a low volume sprint session, and then they go and do a high volume cool down. So when we actually average out all of the adaptations that are happening, what is actually happening here? Is it more oxidative with a dash of glycolysis for the overall kind of response that we're seeing? Is it an oxidative session with a phosphagen source? Or is it just mainly an oxidative session? All right, so my inherent view here, or my hypothesis, if you like, is they're basically just doing an aerobic session with a tiny little touch of sprint training in the middle. So the overarching adaptation or signal for response that they'll see is going to be an oxidative one. All right, and let's see if that holds. So I assembled my Avengers to go and basically go and find out an answer to this. So here's our research team. So we've got Lockie Mitchell over at the VIS, was at the QAS. We've got Louise Cato at the VIS, dietitian. Obviously Lane, Haresh, there he is, yeah. Nathan Lawler, so he's our metabolomics man, he's in West Australia. Maria, who's our epigenetics lady, I'm not going to try to pronounce her surname. Chloe is an epigeneticist at the University of Western Sydney, and then obviously David Pine, who's been around forever and is now retired. He's our swimming physiologist and also immunology. Okay, so I'm going to talk in the next sort of 10, 15 minutes or so about a research project that we did in Queensland. So we flew up, all of us, from 5 different states, which was a nightmare to coordinate. We got there in the end. We did some research over in the Queensland Academy of Sport, as well as also University of Sunshine pool there. So here we recruited 16 pretty good swimmers. All of those were top ranked, at least within 200 in Australia in their event. That included 9 males, 7 females, aged between 16 to 24. What we did initially is we got them all to do a 12 by 25 meter critical speed or critical velocity test. We worked out what their critical velocity was, which was what we then used to go and establish the moderate, heavy, and severe exercise intensity domains for them. They went then and did a session in that moderate, heavy, and severe domain. Now, all that means is they did an aerobic session, they did a critical speed session, so one that's roughly in the middle, and then they did a sprint session. All right. We devised this obviously in consultation with their coach and also with the swimming physiologist that had on board, which was Lachlan Mitchell. So here's what we did. We basically took a capillary blood sample, a milliliter beforehand. They did their swimming session, and then 5 minutes afterwards, we warmed their hand again, and then we took the blood afterwards. Now, just note here, we didn't standardize their food. And the reason why we didn't standardize their food that day, we told them to replicate the same breakfast each time, but generally we couldn't get that over the line in terms of interrupting their training, because obviously they're in a heavy sort of training phase. We couldn't say to them, go and eat something different. That wasn't acceptable from the dietitian's perspective. So that was a bit of a limitation straight out. We then analyzed the blood, both the red cell component and the metabolite component, and the plasma component for The plasma was metabolic phenotyping, and then we also took the red cells and sent them over for epigenetic analysis as well. And what the epigenome is basically really how your genes work, right? So if you think of the genotype, that's basically, sort of code telling you how that gene can be expressed. Then the epigenome is a bit more responsive to that. So that's the layer on top of the gene itself, right? And that can interact with the metabolites to then change how the gene expresses. So that's what we did. There's our schematic. So this part here, that training tells us the metabolite profile, basically how that affects their metabolism. Thereafter, we went through and took their epigenetic profile, and that's the interaction between the genes and the environment. So really overarching here, what we're trying to get at is not only how they train, so that phenotypic response, but then also really what's happening at the genotype level as well. So we know that training is really interaction between genes and the environment. So here we've got an idea of genes and the environment. Right, we're going to talk through a little bit on how data collection went. So this was hard. This was hard. It's one of the hardest data collections I've done until we do Lane's next project. That's going to be fun as well. All right, so the data collection now. Now, we flew in pretty rapidly over in Brisbane after having changed the schedule several times for various different reasons. 5:30 a.m. starts each day. So we stayed in Brisbane. So when we had to go and do testing at QAS, that was okay. We could get up at about, reasonably early, but and go that. But then we had a 5:30 a.m. start at USC, so in the Sunshine Coast. Yet we were staying in Brisbane. Now it's about an hour and a half drive or thereabouts, depending on traffic at that early in the morning. So there was many, many early mornings. I don't know about you, but I'm not that of an early morning man. But basically, it's how it worked. So we took blood. So they put their hand in the bucket. We punctured their finger. We got 1 ml of blood. We then span it down at full depth. So we separated out the plasma and the cells. And then we rushed it to the freezer, froze it in a -80, which we had on site, one at UC, one at the University of Sunshine Coast, and then we took it back for analysis. So this is basically what we did. So here we've got obviously metabolomic analysis and epigenetic analysis. Today I'm only going to talk through the metabolomic component. Two reasons. One, I don't really understand the epigenetics component. That's not my expertise. And two, we haven't finished analysing that part yet. Now, metabolomics, generally the way we analyze that was a combination of LCMS, which is liquid chromatography, that's a separation technique, and then mass spectrometry, which is basically reading the mass to charge ratios, and we break up these metabolites. All right, so we've done that. We then went through and did a multivariate data analysis here. And the approach I'll talk through a little bit more, but this includes things like sparse PCAs and PLSDAs, and what that means, a principal components analysis. PLSDA is a partial least squares discriminant analysis. One is a supervised method, so that basically means we try and separate out pre versus post, for example, without telling the computer basically that there are labels of pre and post. And then we go through and do a supervised approach, which is basically saying, given that we've got a pre-sample and a post-sample, you tell me what metabolites will separate out pre from post. So we give it some help. We give a supervised method. And then we did kind of like a combination of the two of these, which is a repeated measures and over simultaneous components analysis. So it goes from initially, once we've done all of the chromatography, we ended up with 1,640 metabolites. So for those PhD students in the room, that's the equivalent of you having 1,064 Y variables, or if you think of it that way, if you were going to do a univariate approach on this, you would have to do 1,064 ANOVAs and then interpret that. And you wouldn't do that because you'd inflate your false discovery rate and be correct to do it that way. But I'll go through how that works. So here's another schematic on how this works. So basically, I'll just chat through the ASCA approach, because this is one I think that's actually quite useful for a lot of the other projects that I've seen. A lot of like MC's work coming up, she's going to do one of those or has done one of those. If you're thinking univariate, then it's a linear mixed model. If you're thinking multivariate time series, you've got many, many Y variables, then you generally need something like an ANOVA simultaneous components analysis. So what this does is it does the data reduction step and a linear mixed model combined together. All right, so we did all that in the multivariate. We then went through and we did the univariate approach on that. So we did actually write a loop in R, which took a while to run for the 1064 metabolites. And then we did what's known as an upset plot, which is basically a Venn diagram when you've got more than about 5 or 6 different groups. So we're going to spend the next sort of 10 minutes or so discussing through some of those results. So in terms of what we did, we've got time by session interaction. We had a look at that, and then we controlled for sex. So metabolism is generally different between males and females, so it's important to control for sex. Another covariate we could have controlled for was age, but actually the age was quite well matched, so it didn't really need us to control for it too much. So the way this is read, I'll give you the take-home points here. Basically, when we look at this, these are the metabolites, or main metabolites, that were able to separate out the different groups. So be that men and women, or be that pre and post. All right, so the key metabolites that we've seen here are triacylglycerols, or TAG. Now, what a triacylglycerol is basically a fat. So the way fats work is they've got a glycerol backbone, and then they've got some chained fatty acids attached to it. The triglyceride is another name for that. So glycerol backbone is generally something that can then go and be oxidized in the tricarboxylic acid pathway for glucose. And then you've got the fatty acids, which can then get eaten up, usually via something like beta oxidation, and then used to generate ATP. So these triacylglycerols seem to be one of the main kind of metabolites that seem to be separating out the response from the aerobic session that we did, the critical speed session, and the sprint session. We also did this another way. Remember that we've got pre and post data. So we took blood before exercise and after exercise. So we collapsed that time point instead, right? And we did that as a fold change. So that means what ratio from, say, post to pre, or what was the ratio of post to pre, basically. It was almost like a difference score. So a fold change. So what was the fold change between each one of these exercises? So if we have a look at the aerobic, the critical speed, and the sprint we've got over here, you can see there are marked differences in the fold change of these metabolites between each one of them. So you've got aerobic, critical speed, and sprint, and these are the main metabolites that are driving the classification of aerobic, critical speed, and sprint. So it looks to be the free fatty acids, as well as the triacylglycerols, seem to be the main kind of metabolites that are driving the different classifications of each one of these exercises. Now, this kind of surprised me a little bit because we often think, obviously, with this sprint metabolism that we've got there, you would expect to see things maybe that are a bit more like high energetic pathways. So maybe things like, okay, some of those byproducts of glycolysis, etc. But we seem to still have fatty acid metabolism and triacylglycerol metabolism. Now, what I've not shown here is I do have the individual session data as well, and it's pretty much the same story or a similar story for each one of these sessions. So coming back to our original hypothesis here of, is the sprint session reasonably fatty acid sort of metabolism? Is that more oxidative than we thought? I think we can be reasonably confident that it seems that way. So we've got a few bits that we need to do. Still, we've got to finish up some of the bioinformatics here and add on the epigenetic layer. But my initial kind of concluding thoughts really is the physiological stimulus from the sprint and the critical speed session may be more oxidative than initially thought, but we need to confirm that. So we've got to kind of consider what that training soup aspect is. The other thing we need to consider as well is the timing of sampling. We did that about 5 minutes after exercise. I think that's a key component. I'll explain why in a moment. I might just change up one of those slides in terms of, actually, that would be all right. Okay, so why have we seen a lot of fatty acid metabolism in the way that we have? Well, basically, the way lipid metabolism works in the body, we've obviously got adipose tissue, so that's our fat cells. So we've got here or over here our white adipose tissue. We also have another main source of fat, which is our intramuscular triacylglycerols, or IM tags. And then we've got the plasma as well. So what we can do is we can have transfer of fatty acids from adipocytes, so from fat cells, into the blood. Usually they're shuffled on something like albumin, which is a protein, and then whiz around the blood. Then they can then get taken up by the skeletal muscle and used for fuel. Another way this can happen is there's a lot of fat cells within the muscle itself, so these intramuscular tags, they can get broken down and used for energy. Now, athletes are quite different in the way this works. So here's the fatty acid response that we typically see post-exercise. Plasma fatty acids, this is pretty well known, they tend to increase after exercise. And part of that is to basically repay that energy deficit that we have after exercise as the glycogen has been depleted. Thereafter, the glycerol, as you can see, that is going to trend on down as well, doing the same sort of thing. So here we see fatty acids mobilized via the breakdowns of triacylglycerols, diacylglycerols, monoacylglycerols as part of that fatty acid metabolism. And that's to help, as I said, that energy replenishment. Now, athletes, they are a little bit different here before I fall over for myself. All right. Athletes are different. So typically what we see is in the skeletal muscle. All right. So if you've got somebody who's obese or somebody that is insulin resistant, they tend to store a lot more fat in their skeletal muscle than typically you would see. Now, when you've got somebody who's obese, deconditioned, do exercise, even though they've got a lot of fat in their muscle, they tend to mobilize a lot of the fat from these white adipose tissue that goes through into the blood. So you'll see a different fatty acid response. Now, athletes are built different, as I said. So they've got a slightly different structure of the fat. They tend to use a lot more of those intramuscular tags to mobilize fat so that it can be delivered effectively to the skeletal muscle. It's also processed and packaged up in a different way in the tissues as well, such that, as we've got here, we've got different lipid partitioning and localization. So it's stored closer to the mitochondria, so it can be oxidized more effectively. And they have different, what's known as lipid tethering. And so different ways of basically tethering fat molecules so that it can easily get pulled into the mitochondria for oxidation. Now, here's the implication of that sampling time point that I was discussing earlier. So this is roughly how we've measured. So this is from a recent multi-omics study that we've done. We didn't do, sorry, I'd love it if we did this. This was Butte work. This person did transcriptomics, they did lipidomics, they did genomics. And basically what they've done is they've mapped that post-exercise response. You see fatty acid metabolism peaking initially. All right, so generally lipids are active as well as the inflammation pathways. Fatty acids are also involved in inflammatory pathways as well. They're part of that. Thereafter, these fall away, and later on, we actually then end up with the amino acids kicking in later on. And that makes sense because muscle protein synthesis or resynthesis post-exercise, that tends to peak sort of between 2 to 5 hours after exercise. So in terms of what we're seeing here is we're seeing that initial mobilization of fatty acids from exercise and then remaining high as the hormones fall away and adrenaline is still elevated here. As that falls away and the body goes into repair mode, we'll then see a slightly different kind of energetic response or metabolic response. All right. So pretty much a summary and reflection here in terms of this work. Capillary blood sampling is basically an easy method. All right, so we can get that on the pool deck. Blood separation is still pretty annoying though, because we've got to spin it around. Turnaround times are still slow, so it's still months for analysis. This will get better though, right? And also if we change the method from capillary blood sampling to dried blood spot, we could send it in the post. So it's much better. I was surprised that lipid metabolism metabolites were more abundant in the blood than I originally would have thought in a sprint exercise session. It would be amazing if we could do another study that looked at lipidomics with some stable isotope traces in there. So what you can do is you put in a stable isotope, you can map the glucose going through the energy pathways, as well as palmitate here, which is a lipid that's in the body. And that would allow us to work out really the oxidative kind of lipid metabolism, as well as the like carbohydrate metabolism as well. And then there's a whole host of other methodological work that we need to do. I might leave that there in terms of time, but there's one other slide that I did want to show.